All right. Looks like people are still signing in, so we'll give people a few more minutes. Okay, I think we'll go ahead and get started. Welcome everyone to the third webinar in the Rapid Formulation Developments webinar series. The recordings and slides from the first two, the target product profiles and solubilization selection slide, are on our website. So if you missed those or want to see them again, feel free to go to our website and take a look at those. Just a few things about FreeThink. So FreeThink Technologies is the leading laboratory for solving complex stability and formulation challenges using world-class science to speed products to market. Our mission is to use world-class science to speed better products to market. And some of the aspects of that are using our ASAP Prime stability software, packaging, shelf life determination, and ADAP, which is our formulation development. ADAP stands for Accelerated Drug Advancement Process, which is our way of doing rapid and material-sparing formulation design. It has these four pillars: target product profile, solubilization development, stable formulation development, and controlled release development. And we'll be focusing on the third one today. So this is our information if you want to reach out to us or gather more information. We're on LinkedIn and on the web. So moving on to today's talk, it's Formulation for Stability. Our speaker is Maria Krish. Dr. Krish received her BA in chemistry from Swarthmore College and her PhD in physical chemistry with a focus on molecular dynamics from the University of Chicago. She conducted postdoctoral research in physical chemistry at the University of California, Irvine, studying the surface structure and reactivity of liquids. Dr. Krish served on the faculty and led a research group at Trinity College in Hartford, Connecticut for 8 years before joining FreeThink Technologies, where as a research fellow in physical sciences, she is responsible for leading company projects focused on chemical and physical stability, as well as training new users in ASAP Prime software. So as I turn it over to Maria, just want to let people know that we'll be answering questions at the end. And if you have any questions that come to you as Maria is giving her talk, feel free to put them into the Q&A. There should be a Q&A button at the bottom of your screen. So now I'll turn it over to Maria.
Great. Well, thank you so much, Jennifer, for the introduction, and thank you to the audience for coming today. As was mentioned, FreeThink Technologies has a focus on stability, and so today I'll be talking about how we think about stability in the context of formulation development. So in terms of what we'll be covering today, we're going to talk about the advantages of using what we call a tiered formulation approach in conjunction with ASAP studies and the software ASAP Prime for performing rapid, material-sparing drug product development. I'll be talking about how to avoid some common pitfalls associated with binary excipient compatibility studies that are very common in the industry. I'll be talking about some practical considerations for designing and executing a tiered formulation screen. And finally, how to assess and handle oxidation in the context of a tiered formulation development. I will mention that I'll be using examples largely drawn from oral solid dose development today. That doesn't mean that this tiered formulation approach doesn't work with other dosage forms, but it's an easy first example to use. So I'll be drawing from a lot of sort of tablet development examples or capsule development examples, but for instance, liquid products can also be used very effectively with this approach. And actually, in a lot of ways, some of that design is simpler. So to begin, I'd like to talk in a general way about how we think about the interaction between excipients and a drug product with regard to stability. And the unfortunate truth is that the drug substance is typically more stable than the drug product. So we have a sort of Sisyphusian a job in formulation in that you're trying to make a stable drug product, but you're almost guaranteed that it will be less stable than the underlying drug substance. But why that changes is something we need to think about. So there's, I'd say, two main classes of impact that excipients have on drug products, and those are direct and indirect. So I'm first talking about direct impacts. So first of all, the sort of simplest case to think about is a direct drug excipient reaction. So there's a well-known reaction of this type, the Maillard reaction, between secondary amines and reducing carbohydrates. Usually the culprit is lactose. That's known as a Browning reaction and is seen in some drug products. And so this is often what people think about when they think about drugs and excipients interacting. But actually, this kind of direct reaction is relatively uncommon. So it can happen, but it's rarely the shelf life limiting issue. What's probably a more common direct interaction is the reaction of impurities from excipients with drugs. So there's a number of different types of excipient impurities that are well known to react with certain classes of drugs. So some examples here are things like peroxides, often impact oxidation reactions heavily, formaldehyde, certain acids, especially small acids that might be found, such as formic acid and acinic acid, and nitrites are a common class. And if you caught the nitrosamine webinar series that FreeThink sponsored a few months ago, there was a lot of discussion in that webinar series about the reaction of nitrites and drug products. So while those direct impacts can play a role in stability, what's actually often an even bigger impact is indirect interactions between the excipients and the drug products. So what's going on here? In general, you have increase in mobility of the API as you lose crystallinity. And that loss of crystallinity can happen at an interface. And specifically, in this case, we think about the drug excipient interface. And once you have an increased mobility, you also have an increase in degradation rates. So I like this image as sort of a mental picture of what might be happening on the microscopic level in a drug product. So here you have little bits of crystalline API, and they're clinging to this much larger amorphous excipient, right? And so if you think about it, the environment in the middle of one of those little crystalline API pieces, it's a homogeneous, well-ordered crystalline environment. That looks extremely different from the interface where one of those crystals is touching the excipient, right? So you're getting an interface, you have less order, you're having less structure, and as a result, you have a higher reaction rate. So again, this is an indirect impact. It's changing the reaction rate of the API by a physical process, but this tends to be a very strong effect. As part of that effect, there's a common relationship between drug load and stability. And in general, the degradation rate is inversely proportional to the drug load. So there's several models that have attempted to refine this understanding of this relationship, and they all assume that degradation is accelerated by contact between drug particles and excipient particles. So generally, if you have a lower drug load, you have more of these API excipient interactions, you have faster degradation as a result, and what you get is a less stable product. So again, a lower drug load tends to lead to a less stable product. Now there's an asterisk here. in that dimerization reactions can be an important exception to this relationship. So if your main shelf life limiting degradant is actually a dimer of the API, then you need two API particles to come together. And there are cases where that means that this relationship looks different. There's cases where it follows this and cases where it doesn't. So if you do have a dimerization or some sort of higher order polymerization as your limiting factor, maybe look at that very carefully. But in the common example, you have this inverse relationship. And if you want to dig into what that looks like, there's been a number of papers published in the last 10 years about how exactly this might model. And so I've listed a few of the main ones here. The first two papers end up deriving a power law relationship between reaction rate and drug load. And then the third has something that's fairly similar, but a little bit different, and that's called the surface area contact model. So if you want to dig into any of those three models, the papers are referenced here. But the common theme between them is that all of these models have a strongly nonlinear dependence of stability on drug load. And ultimately, dependence is based on the surface area contact between the API and excipients. And it does not depend on direct reaction between the API and excipients. So again, this is a strong dependence, and it's an indirect effect of excipients. So sort of a demonstration of what this looks like. These are some nice data that were presented in the 2022 SOS conference presentation by Gary Scrivens, who's one of the major authors of that surface area contact model. And what he was showing here was on the x-axis drug load, and on the y-axis percent degradation. And this is experimental data from some published studies where they took different products and mixed them with a common excipient, microcrystalline cellulose, and basically looked at the impact in binary mixtures of changing the drug load on the amount of degradation you measure. And it's a huge impact, right? So if you look in this case over here for product C, if you're going to 100% drug load, you basically are looking at the API. So you're here, you have about a 1% degradation. As I decrease the drug load, increase the amount of excipient, I get to about 50%, still look mostly like the API. Down to 20% drug load, starting to grow a little bit. And then when you hit, in this particular example, about 5% drug load, and you keep going down, you have just an enormous dependence on the drug load, right? So between 5 and 2, you have, more than doubled your degradation. So this is a very strong relationship, and you can see it varies depending on the system, but in every case, there's a strong drug load dependence. Okay, so I'd like to take that understanding of how excipients can impact drug stability and think about one of the very common first steps for a lot of evaluation of excipients for inclusion in formulations. And that's the binary excipient compatibility study. So the approach for these is usually that a drug is mixed in a binary combination with a whole series of a panel of potential excipients, right? Each binary combination is separately stressed and tested, and typically this is for degradant growth, but if there's a different shelf life limiting characteristic of interest, it can be for something else. The results from all of those binary mixtures are then used to pick the acceptable excipients and used to inform development of a full formulation. So first you do the panel of binary excipient studies, then you use that data and that understanding to build the full formulation. So how does one go about designing one of these studies? Well, one of the first things that you need to choose is what ratio are you going to make these binary mixtures, right? So we're dealing with the drug excipient ratio, and you have to choose something, right? So you have two things. A 1-to-1 ratio is actually the most common ratio that's used for these, right? So that means 50% drug load. It's simple. You can pick it for everything. But there's also ways to do this where you try to do a representative ratio. But the question then becomes, what is a representative ratio. And this is actually difficult to uniquely define. So to take an example, if you have an API at 1% drug load and an excipient at 5% drug load, the question is, what is a representative ratio to use for this binary mixture? So you could make a good argument for saying, okay, a 1:5 ratio is representative, right? Because that is representative of the ratio of the API to the excipient in the larger formulation. But you could also say, well, the excipient is only present at 5%, so 95:5 is representative, because in this case, the excipient is at 5%, and that's what it is in the formulation. So within the study design of binary excipient compatibility studies, you could get a 1:1 ratio, a 1:5 ratio, or a 95:5 ratio in this particular example. And this is important because you have, as we've seen, a very strong drug load dependence. So just to put that in terms of drug load, a 1:1 ratio is a 50% drug load. A 1:5 ratio is a 16% drug load, and a 95:5 ratio is a 95% drug load. So I want to zoom back to these graphs for a minute. Imagine you had the case on the left here, right? So if you look at a 95% drug load on this graph, that looks like a pure API. So your reaction rate in that case will look pretty much like the pure API if you're looking at indirect effects of this excipient. If you go to 50%, you've increased a little bit. It's probably measurable increase, but it's fairly modest. If you go to 16%, you've either tripled or quadrupled your degradation rate. And again, this is not from a specific reaction of the API with the excipient. This is from an indirect behavior that's based on the ratios you're using. So the lesson here is that the results you're going to get from a study like this is highly dependent on how you choose to mix the ratios of excipients, and it's going to depend on whatever ratios you decide to test. Another thing that can happen with these studies is that if you have excipients that are themselves at low levels, you can sometimes exaggerate issues with them. And the most common example of this that's been seen is that 1:1 API magnesium stearate binary studies have led some companies to exclude using this lubricant. So there are certain reactions that can happen with this, but they're often no issue if they're used at low levels. And magnesium stearate is typically used less than 1%. It's often 0.5%. And so this is something where you have an excipient that may actually be fine at the target level, but any small issues may get exacerbated in a binary excipient compatibility study. Ultimately, the sort of take-home lesson from this is excipient effects are concentration dependent and they're nonlinear. So the binary impact is not directly additive. So you can get a lot of information from the binary compatibility study that really changes once you've made the target formulation with all of the excipients at their target levels. Okay. So these are some issues with binary excipient things. And then I'd also like to talk about how we evaluate data. And this data can be either from binary excipient compatibility studies or from full formulation studies. And this is the question of rank order screening. So this is a question of once you have some testing of formulations, how do you test them? What are you looking at? So a common way to quickly assess either binary compatibility or full formulations is to look at a single very accelerated condition, again, to get data rapidly and to say, okay, how does this formulation or mixture perform at a highly stressed condition? So an example of this is two formulations. And in this case, I'm going to look at full formulations, not binary mixtures, stressed at 70 degrees C, 70% RH for 2 weeks. And so when this is done, you get some results. One of these formulations gives you less than 0.2% degradant. So this is below most unidentified degradant specifications. And one of these formulations gives you 0.9% degradant after 2 weeks. And so the ultimate question here is, which formulation do you proceed with for 25 degree C, 60% RH storage? So if this is the only data that you have, you're looking at this, one of these clearly reacts more, right? One of these has much higher degradant levels. And so if you have one data point, you would rank formulation A as better than formulation B, right? It seems like a no-brainer question. But the issue is, can you get sort of an unrepresentative picture from having one highly stressed condition? Because remember, you're not actually storing this at 70 degrees C, 70% RH. You're storing this at 2560. You're trying to use this accelerated condition as a tool. So what is that accelerated condition telling you? Let's see what happens when we get a little bit more data on this system. So if instead of looking just at 70, we look at 70, we look at 60, and we look at 50, we get a fuller and different picture of this system. So what I have here is what's called an Arrhenius plot. So this is the natural log of K versus 1 over T. And you would generally expect linear behavior for kinetics. So we see that. We see some lines. So we have both of these formulations tested at 70, tested at 60, and tested at 50. And if you look at this, formulation B has higher amounts of degradation at every tested point. So formulation A continues to have lower degradation at 70, 60, and 50. But there's something important that's different about them that's not captured in that one point. And that's that the slope of the change is different. So B has higher amounts of degradation, but it also has a higher slope. Whereas A, with its lower amounts of degradation, has a lower slope as well. And that slope is called the activation energy, or the EA of a reaction. That's the temperature dependence of this reaction rate. And why is that important? Again, what we're interested in is not behavior at 70 degrees. That's just a tool. We're interested in 25-degree storage. So what happens when we follow that slope back down to the storage condition? Those two lines cross, right? So when you go back down to 25 degrees, it turns out that formulation A, which changed more slowly with temperature, is actually predicted to have higher amounts of degradation. And formulation B, which had a lot at the accelerated condition, is actually more stable for room temperature storage. So it's important, if you're thinking about rank order, to realize that one condition at an accelerated condition will not necessarily give you the rank order that you're interested in for room temperature conditions. And in fact, in an example like this, it can give you the opposite order of what you're interested in. So it's important to think about understanding the behavior at storage conditions and using things as a tool that gives you a picture of the storage condition. So this can be an issue either whether it's a binary excipient assessment or a full formulation assessment. Again, your focus should be on the storage condition. So how do we do this? So I want to talk about moving beyond sort of rank order at one condition. And the approach that FreeThink uses is ASAP. So we want to understand and model storage behavior. And essentially what we want to do is we want to understand those slopes. We want to understand the change with temperature. And it turns out that a lot of formulations are also sensitive to relative humidity. So we want to understand the change with relative humidity as well, slope. So we use what we call an Accelerated Assessment Stability Program, or ASAP for short, and a software program, ASAP Prime, that works with that to model that data. So what does this look like? So in order to perform a study to try and tease out these slopes, what we do is we stress formulations or a drug product or a drug substance at several temperatures and open RH conditions. And the key idea is to measure the amount of degradation over time at each of these conditions. So we basically are interested in two variables, temperature and relative humidity, and we want to make a model in those two variables. So from a practical point of view, this looks like putting a drug product in a nice closed container. We set the relative humidity with a saturated salt solution, close it up in a little ball jar or a canning jar, and we can put that whole system in an oven and have a elevated temperature, set RH condition, leave it for different time points. Once we do that stressing and we measure whatever shelf life limiting characteristics are evolving, we then calculate the time to fail at each of those conditions. So we want to know the failure time. And again, that's because from a pharmaceutical point of view, we're interested in failing specifications or passing specifications, and we want to understand how long it takes to fail. So we get a time to fail as a function of temperature and RH, and that's our core data set. We take those times to fail, and we fit that data to what we call a moisture-modified Arrhenius equation. So the first few terms here should look familiar from chemistry classes. This is the Arrhenius equation, not new science. Looking at the rate dependence, you have a pre-exponential factor here. This is, again, the temperature term, where the activation energy is the slope of the temperature dependence. And then there's also a moisture dependence for solid products. So you have RH as a variable, and this factor B is the humidity factor. So we fit all the times to fail to this equation. We get a model, and that model is used to predict time to fail at the storage condition. So the general approach in terms of thinking about how to use this for stability is what we call a tiered formulation screen. So we take a full formulation where everything in the formulation is present at representative of the full formulation amounts, right? Now, this doesn't necessarily have to be the complete final formulation. So often, instead of a real tablet, it would be a compact, it might be a blend, but you have all of the excipients present and you have them in the right ratios. We conduct an ASAP study with this temperature and RH as variables on all of the full formulations. And from each of those studies, we build a predictive model that gives product shelf life in representative packaging. And again, this model has two variables, temperature and RH. So I think of the role of packaging as basically modifying RH, right? So your temperature is set by your climate zone, but your RH can be modified by your packaging. If you're doing an overall rough screen, one thing you can do is assess the data instead of the specific packaging, just look at low and high RH to mimic very protective and less protective packaging. Or you could do specific packaging and calculate the impact of that. Once you've assessed this full formulation, then it's a good idea, and I'll talk about this more later, to optimize and retest your final formulation. So again, what our goal is we want a model of product stability in representative packaging at storage conditions. And we want that model to work for the full formulations. So the way we think about these tiered formulation screens is sort of visualized here, right? So tier 1, we make 4 to 6 full formulations. We prepare each of those formulations and perform an ASAP study on them. Hopefully, at least one of them will be stable at the target shelf life. If that's the case, we would then recommend optimizing that formulation and testing it again. If none of them are stable, hopefully you got some useful information from the first 6 or 4 that were not stable. You can reformulate, make some changes, and do tier 2 of some new formulations. Again, prepare those and perform an ASAP study, and hopefully one of those will be stable enough to go forward. The fundamental result that we're looking at from the screen is whether something is unacceptable or acceptable for the targeted storage condition. And this can actually, depending on what the target product profile is telling you about what's needed. And for those of you who missed Jessica Kelly's lovely webinar on target product profiles, that's available on the website. But that can be a useful way to think about what you need as your targeted storage conditions. So if you need several things, once you have a model, you can actually evaluate different climate zones that have different temperatures, different external RHs. You can also evaluate different representative packaging options. Again, the major role of packaging is to modulate the internal RH. So if you have RH as a variable in your model, you can understand the impact of packaging. One possibility is that multiple formulations are acceptable. And in that case, you basically can choose your formulation based on orthogonal characteristics that are not stability, right? So in that case, you want to look back to the target product profile and select based on something like manufacturability or biological considerations. So what are the next steps? So in most cases, I would say that the tier 1 develops some formulations that can advance. If it doesn't, the next step would be to go to the tier 2. But even with a second round, these formulations are relatively short timescale for development. Once you have some formulations that are both acceptable for stability and are your choice for other reasons, generally there's some refining that happens. And we would highly recommend reevaluating the stability once that's happened. And there's a few reasons for this. One of the big ones is that early API lots tend to vary quite significantly in stability. Now, the good news is the variation is usually improvement, right? So typically the earliest API lots are dirty, they're less stable, maybe less crystalline, and they get refined over time. So if you do an initial formulation screen on a very early API lot, and then that API lot changes significantly, it's worth redoing the screen with something that's representative of what you're moving forward with. It's also the case that you may have changes in things like excipient grades that have been chosen or processing methods that may impact stability. And maybe they seem like minor changes, but it's a good idea to make sure that that's a correct assumption. So if we're thinking about designing one of these screens, what kind of considerations come into play? So first of all, it's useful to have a control test of the pure API. And this gives you kind of a baseline stability for the formulation. Again, the formulated drug product is almost always less stable than your pure API. So it's nice to know sort of what is your limit. And I will say in the few cases I've seen where the tier 1 formulation did not meet the stability target, it actually turned out that the pure API also didn't meet the stability target, right? So you were dealing with an API that itself did not meet the target shelf life. So it was not a surprise once you realize that the formulated API was failing. There's some potential characteristics that you might want to cover if you're considering including them in the formulation. So for instance, you can compare the approach of wet and dry processing. Or if you think that there might be a pH dependence of the formulation, you could add acids or bases to the formulation and try and understand if that has an impact. The choice of API drug load is also very important. And again, this goes back to all the discussion about the drug load dependence that we started the talk with, right? So the drug load will vary, will impact your stability. And so you want to make a good choice of drug load. So again, this is a case where you can go back to the TPP. If the dose is set in the TPP, then absolutely you would want to use the target drug load. Now, it may be the case that the dose is unknown, and in which case you want to look at the lowest likely drug load. You want the lowest one because to some degree you want the most conservative case. So you want to choose the lowest one that will show the highest amount of degradation. This also means if you don't get adequate stability in that formulation, one way of improving it is to increase the drug load, right? Because if you get something that's on the edge of stability and then you increase the drug load, you're likely to find a stable product. And then depending on what you know about your API, there might be specialized characteristics of concern. So an example is things like compression have a very minor effect on many formulations, but occasionally you will find a case where it's important. And so if you had some indication that that might be important, you might want to include a comparison of, say, a blend and a compact. So here's an example of what a formulation screen might look like in terms of types of formulations that are tested. So here I have 4 formulations listed out and all of their components based on the function. And what you can see is within this formulation screen of 4 formulations, I have some variation between the brittle diluent, the disintegration, the disintegrant, the binder, and the lubricant. And there's also 3 formulations that have the same drug load and then 1 where it's been varied a little bit. So these are the types of things that you can vary in these formulations. And again, any of these formulations are reasonable formulations that you might be able to move forward with if they have adequate stability. So what kind of results do you get from a screen like this? So this is sort of an example table of results. So here we have 5 formulations. And in this case, I'm starting with formulation A, which is sort of the base formulation. Formulation B and formulation C have both had alternate diluents chosen. Formulation D has been made in an acidic environment, and formulation E has been in a basic environment. And so what we're evaluating here is basically 2 cases. We have 25 degrees in both cases, but then we have 35% RH, which would be used to mimic protective packaging, and 60% RH, which would be used, it's essentially open, to mimic unprotected packaging, right? And so if you look at these results, this is the probability of passing after storage for 2 years. And different companies have different acceptable levels. What we would usually recommend is to look for at least a 95% probability of passing. So what you can see here is that 2 of these formulations have met that criteria. Formulation A, which is 95 or 97, depending on the humidity, and formulation D, which is 100% in both cases. So either of these formulations could be reasonably advanced for refinement and retesting. The other formulations, don't look all terrible, but they don't meet the 95% probability of passing. And notice that this does give you some sense that the pH in this particular formulation probably matters, since adding acid made it better and adding a basic environment made it the least stable of the options. So some ASAP study considerations, and this is especially for folks in the audience who might be familiar with ASAP studies from other contexts. For a formulation screen, we typically recommend designing a slightly smaller study than usual, because the real goal here is stability bucketing, right? You want to know if something has adequate stability, but there's an understanding going into the screen that there's probably going to be some tweaks to the final formulation. So you have something that can be moved on to be advanced, but for instance, it might be a compact and not compressed as much as a final tablet. It might not have a coating. So there's all sorts of things that might change a little bit in the final formulation. So from just a material use point of view, speed point of view, it's helpful to have a slightly smaller study. So a typical study might, something like 22 samples. It depends a little bit on the other considerations. To test the final formulations, then you would want a larger study for a really refined shelf life estimate. So once you've tweaked everything, once you're sure of the drug load, you're sure you're using an appropriate API, that's when you tweak it and have a very large study. And this also helps minimize API use in the early studies. It's also the case to keep in mind that the shelf life limiting degradant may vary by formulation, right? So in this particular graph, I'm just telling you the probability of passing. I'm not saying anything about what limits that probability, but it's entirely possible that the degradant that's limiting formulation C is different than the degradant that's limiting formulation A. So I'm just saying all degradants pass their specification with at least these probabilities. But it's important to understand that as you change the formulation, you may have different degradants become more or less important. And what that means from a practical sense is you do want to consider all of the degradants that might be growing in a formulation upon stressing. So what are some of the advantages of this approach? Well, first of all, you get information beyond rank order, right? So you really get an acceptable or unacceptable result. So if you have rank order, you have this one's better than that one, but you don't know if all of them are good or all of them are bad. And those are 2 possibilities, right? You may find that no matter what you've done to the formulation, you're stable. That's great. Or you may find that it doesn't really matter which one is better because none of them are good enough to meet your shelf life. And in that case, the understanding of which is most stable may be helpful for the next step, but you can't go forward with that. It's also a material sparing approach. So because you're skipping the entire binary excipient compatibility and starting with full formulations, you really have an opportunity to minimize the API use. It, of course, depends on the dosage strength that you're using. If you're using very low dosage strength formulations, like 1 or 2%, it's often the case that under 1 gram might allow you to do the full study. If it's a higher dosage strength, it might be a little bit more. And it's a rapid approach. So it depends how many tiers you have, how long it takes, but a tier 1 study length is usually about 8 weeks, right? So you can get a formulation that you can move forward with, or maybe several, within 8 weeks. So we've discussed sort of the approach of a tiered formulation development. And I'd like to talk about a specific type of stability issue that fits very well in this kind of approach. And that's oxidative degradation. So this is a kind of a specialized case, but it fits well. So I wanted to talk about it in a little bit more detail. And I'd say that broadly, there's 2 main categories when you're thinking about oxidative degradation. One is a case where you don't know if it's a problem. So you really have no prior data and you're wondering, is oxidation something I need to be concerned about? And the other is where you have some prior knowledge and you know that oxidation is a concern. So if we think about the first case, the role unknown for oxidation, it's actually pretty straightforward to include a sample comparison in your formulation screen. And so what you would do is you would take a sample that's going to be stressed at the same temperature and RH stress condition, and you remove the oxygen from one of those samples and leave the oxygen in the other. So there's lots of ways to remove oxygen. You can flush it with nitrogen. You can put an oxygen absorber in. But basically, you want the stress condition to be the same except for the presence or absence of oxygen. So if the degradation amount responds to the oxygen level, that's a pretty good indication that you probably are dealing with an oxidative degradant. And I should mention that these screens are also not only telling you about degradant growth, but they're telling you which degradant is going to limit shelf life. So it can specifically indicate if the shelf life limiting degradant is oxidative. So it could be that you have some oxidative degradants, but they don't limit shelf life and they're really not a major concern. But if your shelf life limiting degradant is oxidative, that's when you want to start thinking about limiting those oxidative reactions. So the second category here is when oxidative degradation is a known issue. So this is a case, maybe you had some data from formulation screens, maybe you know something about the chemistry of the API, but you have some reason to believe that oxidation is a problem. And this can even be a case where you have a formulated product and you have sort of oxidation that's out of control. So you could actually do a screen where the entire point of the screen is adding control for oxidation. So the simplest way to approach this is to include formulations with antioxidants in the screen, right? So one easy way to get rid of oxidation, if it works, is to have an antioxidant in the formulation, and then you've solved the problem from the beginning. So if you're assessing antioxidants, generally we would recommend using a relatively high percent of antioxidant to assess qualitative impact, right? Because again, in this case, you're trying to understand, does the antioxidant work? And part of that is it using the right mechanism of action? So there's a wide variety of different antioxidants, and they really use different mechanisms. And that means they target different types of oxidative reactions. So for example, you could include in a screen antioxidants that are chain terminators, ones that are sacrificial reductants, or ones that are chelators taking up metals. And so these will address different types of oxidation, and some of them may work and some of them may not. If you'd like some more detail on thinking about oxidative mechanisms and the mechanisms of antioxidants, Ken Waterman has a really nice review paper that I've listed here that talks about oxidation in general and approaches to minimizing oxidation. So once you figured out that you do have an oxidative degradant that's a problem for stability, what options do you have to fix it? So first of all, if you did an antioxidant screen, that's great. Hopefully you've identified an antioxidant that works, has the right mechanism. And at that case, the next step might be to optimize its level in the formulation. So to some degree, you may think of that as a second tier. So you often start with a high level, and you might want to reduce that and see how low you can go and still have it work effectively. It's also the case that certain excipient impurities have been known to cause oxidation. So a common culprit for this is things like peroxides. So these are often present in excipients. They can also grow in excipients. And so if you have a way to minimize those, that may help an oxidative degradant that's limiting shelf life. If you're still at an early stage of development and different forms of the API are being considered, it's worth considering protonation of the API. So in general, oxidations are hindered if the API is already positively charged. So again, if you're still doing API screens, that's something to think about. And that's something that can, again, sort of stop the problem at its source. And finally, if you're having limitations for whatever reason and actually solving this through formulation, this is a formulation development focused webinar, but it is worth noting that there are other options. So oxygen protected packaging can be very helpful if you, for whatever reason, are using a formulation that has an oxidation problem that you can't really fix. So this is usually a combination of things. One key component is low oxygen permeability packaging. So basically, you don't want oxygen continually coming in because it's always going to be at high levels on the outside. And then you need something in the package to reduce that oxygen level. Now, if you have an oxidative degradation going on, the product itself may reduce the oxygen level just by reacting. So it's possible that all you need is a low permeability packaging. But there's also more active ways of reducing the oxygen. Specifically, oxygen absorbers are very effective and very quick, or flushing with nitrogen. So you use some way to get the oxygen inside packaging low, and then the packaging, if it's protective, will keep the oxygen from coming back in. So again, these are ways to think about specifically dealing with oxidative reactions in the context of a formulation screen. And as I said, there may be cases where you actually start with a full formulation and the screen is really adding antioxidants to it. So with that, I think we've come to the end of the talk. So just to kind of summarize the key points that hopefully you'll be taking away from this, we talked about using a tiered formulation approach in conjunction with ASAP Prime for rapid and material-sparing drug product development. And the key here is we're focusing on full formulations. So if you find one that's good, you can sort of immediately move forward with it. And you're understanding its behavior at the storage condition, not at some accelerated condition. We talked about how to avoid common pitfalls associated with binary compatibility studies, specifically looking at the drug load dependence, which can be quite extreme, and looking also at understanding the behavior rank order and making sure that if you're rank ordering things, you're dealing with behavior at the storage condition. And we talked about some practical considerations for designing and executing the tiered formulation screen, and finally, how to assess and handle oxidation. And with that, I am happy to take any questions. Turn it over to Jennifer to moderate.
Great. Thank you, Maria, for a great talk. We do have some questions here. Let's see, we'll start with regarding sort of excipient compatibility studies. So one of the things you talked about was using this ASAP approach and tiered formulation approach to replace binary excipient compatibility studies. Has that been used in regulatory filings, and has that been accepted as a substitute for binary excipient studies?
Yeah, you need to justify the formulation, but that justification can happen in a lot of ways. So it definitely does not have to be through a binary excipient compatibility. And as we showed, sometimes those binary excipient compatibilities don't necessarily give representative answers. So yes, it has been used, and there's no regulatory requirement for having a binary excipient compatibility study as the way that you're evaluating the formulation.
Great. And you mentioned something about blends versus compacts. Can you talk a bit about what you generally use, sort of for the compressed version or non-compressed version, and what that might be, where you might use the different ones?
Yeah, so again, this is a fairly rapid approach. So there's often the formulations aren't going to be the final version. So one way to do that is to just not compress them at all and use a representative blend. So in that case, you'd have every component present at representative combinations, but just use it as a blend. There is usually some dependence on compression of degradant amounts. So basically the amount of contact is somewhat related to the density and the compression, the solid fraction. So using a compact that's somewhat compressed is often a way of getting at that. Again, that's one of the reasons why for the final formulation where you have a fully formulated tablet, you might, even if you did complex early, you might want to retest. But the compacts are often a quick way of assessing that there's been some compression. So it's a little bit closer to the final formulation than the blend, but fairly quick to develop. And I would say it's fairly uncommon that compression is a major effect, but I won't say it never happens. So if you know that that's an issue, that's when you might want to do a comparison.
Okay. And you talked about other types of dosage forms. So presumably you could use this approach for something like liquids, and how would that differ from how you do it for solids?
The liquids are actually easier because when we're thinking about testing, we're doing models with, in all the examples I gave, two variables, so temperature and RH. But if you have any liquid with any kind of aqueous component, you don't need to worry about RH, right? So you only have a model with one variable, temperature, and that means you can actually have a smaller study because you only have one variable to model and one variable to vary. So to some degree, it's actually a smaller and quicker study. You basically just use a lot of different temperatures to stress the liquids. But other than that, other than not having to deal with the humidity component, in that case, it's fairly similar.
Great.
And
Okay. And then there's a question about, does this strategy apply to evaluating nitrosamines as degradants?
it can. So I think, so you need to test for whatever is limiting stability. So if your shelf life limiting characteristic is growth of a nitrosamine, then basically you would stress these formulations and measure the growth of nitrosamines, right? So obviously that's usually a different analytical method than the rest of the degradant growth, but basically one thing that you might do, and people definitely do, and we have done, is to measure those formulations with regard to the regular HPLC or UPLC method, and then also have a separate method that's targeted at nitrosamines. I would say that if you go back to some of, FreeThink ran a webinar on nitrosamines a few months ago, and there was some interesting ideas about trying to mitigate nitrosamine growth. And so some of those ideas actually can be incorporated into the formulations that you're testing in the screen.
Okay. And there's a question about evaluating different packaging options for this. How do you determine what different packagings are? And then if there's a particular packaging that's of interest, what needs to be measured or sent, and kind of what kind of experimental data do you need to evaluate that packaging?
Yeah, so the beauty of having a model that has RH as a variable is that, as I said, packaging, the major role, in my mind, I'm focused on stability, but in my major role of packaging is really to modulate that RH. And this is something that's really easy to calculate the impact of packaging. So basically what you need to know is you need to know the moisture vapor transmission rate of your packaging. And you need to know sort of the starting water content inside and things like that. But basically that's what you need to understand. You need to understand how quickly your packaging is letting water in, and then The other thing you would need to understand is the product itself usually holds some of that water. So you need to characterize how the product takes up water. And that's like a moisture sorption isotherm. So the general idea, though, is this is something that's pretty easy to calculate. So once you have a model of temperature and RH, you can pretty much do a calculation to assess different types of packaging. So say you wanted a blister package, the major difference between the blisters is that they're going to have a different moisture vapor transmission rate. And so you can calculate what the RH will be inside that blister based on the different moisture vapor transmission rate. And then again, this is something where in terms of looking at what options are reasonable, you might look at your target product profile and see what are the requirements for packaging.
And then another question, is it typical to use open or closed containers for high relative humidity screens? You mentioned closed in one of the slides, so is that always the case?
So what I would say is that the product is open to that RH. So that's very important. The product itself is not in packaging. And that's super important because RH is a variable. So we want RH to be a constant variable in these studies. That being said, it's much easier to set the RH in a little canning jar than it is to set it in an entire oven. Now you can do both, right? Most companies have stability chambers where it's set, but it's much easier to set it in a small environment and allows you to do lots of different conditions. So our particular choice at FreeThink is we make basically little closed containers at constant RH. Some companies that use the ASAP approach will use set RH ovens. As long as the product that's being tested is exposed to constant RH, either approach is fine. We just find this practically a lot more convenient.
Okay. And then can you use this ASAP approach when the degradant further degrades into something else? Because that would sort of talk about something that, how does that affect like your modeling with that number going down?
Yeah, so secondary degradation is definitely a concern for some products. And there are ways to use the ASAP approach. Sometimes the analysis is just as simple as it is for a primary degradant, and sometimes it's a little more complicated. So it's a little bit more case by case because it really depends. If you have a series of reactions, it depends on what the relative rate constant is. So in some cases, you may go straight through the first reaction and basically only be modeling the second degradant. And in some cases, there's more of an equilibrium. So it's a little bit more case by case, but it's absolutely something that does come up and is modeled a lot. And FreeThink has given some talks on that specific topic. Christina Flavier has given some. I think they're on our website, though I'm not 100% sure. So maybe don't quote me, but I think they're on our website. So that it's a good question, and there's a little bit more depth in there, but it absolutely can be used.
Okay, great. So I think we're out of time. Thank you, Maria, for this. Just a reminder to folks that the slides and the webinar will be up on our website shortly. So if someone else wants to take a look at this would be great, and we'll address any of the questions that we haven't gotten to offline. But thank you all very much. And we'll see you next week for Sherry Shamblin's talk on controlled release.